and I'd like to thank all four speakers for giving us such an insight into their work. So I've got a few questions for you each in turn, if you don't mind. Firstly, though, I'd like to add just one statistic. So Andrea took us through some very powerful information regarding the cost of dementia and the burden that dementia does create now in the world. Another statistic, we've reached an inflection point. So in 2015, the number of people aged 60 to 64 years now exceeds the number of people aged 20 to 24 years. And so the number of people working and paying taxes to care for people like Tony and me as we get older is going to be a real problem for society, I think. So, Elena, firstly to you. One of, I think, the observations made on the early transplants in Parkinson's disease was that the transplanted neurons also suffered from some of the pathology when they were put into Parkinson's disease. There was evidence of alpha-synuclein deposits. So in terms of their utility, for example, in Huntington's, do you think they will buy the patient more time but eventually succumb? Or because they won't have the Huntington gene, you know, the triplet repeat, they will be spared? What's your view?
Well, I mean, if you think at the results in the Parkinson field, I mean, the colleagues working in the field, so basically they suggest that this transfer of alpha-synuclein is interesting biologically because it may tell you something about the disease. But I mean, to me, they don't seem to be so worried about the transplanted neurons suffering, I mean, from, this transfer of a pathological protein. So, and also they have data showing that even 15 years after transplantation or the initial transplantation study, so the fetal cells, fetal mesencephalic cells transplanted into PD patients, so 15 years after transplantation, yeah, you have cases, this was reported, of a patient, so this was postmortem material in which the donor cells had acquired the alpha-synuclein, yet those patients and those cells were still alive. I mean, the patient were still gaining benefits and the cells were still alive. So in Huntington disease, let's say the story is just beginning, so there are a few papers showing that indeed there are mutant huntingtin aggregate protein, being transferred from the host to the new cells, that are being transplanted. There are a few papers in this direction. Let's say, but probably, the situation is as for Parkinson's disease. Now, it takes many years to develop symptoms. And my impression is that if we are able, as for Parkinson's disease, to provide the brain, with new cells, we may give more time to the patient. And I think this is really a goal that we should be able to face and fulfill.
Yeah, and you mentioned something very interesting there, which is that many of these diseases are diseases as you get older. And one of the features, as we know, of stem cells, and you articulated the wonder, the beauty, and the fascination of working in that field, but they age in the Petri dish at the same rate that they age in people. And so when you're dealing with them and using them as experimental models, you're actually dealing with very, very young neurons. Would you like to just comment how the field is trying to address that technical issue?
Yeah, I would say that, probably we are dealing with very young, I would say beautiful neurons. We are at the beginning. There are suggestions from other groups, like Lawrence Studer again, on how we could try to, change the age of these neurons in vitro, for example, by working with the progerian protein. And, but let's say, we're studying them from a functional point of view. And they seem to, give rise to, they seem to reveal interesting functional properties. So although they are probably young, but they express a repertoire, or they seem to express a repertoire of receptors and neurochemical properties. We're digging into this that, give us some hope, in terms of understanding more about their biology. But definitely being young or old, is something that has to be further study.
Okay, thanks. Coming onto you, Andrea. You're obviously taking a very innovative approach here in trying to find anti-A beta antibodies, and you showed some of the very promising clinical data that we've received. I guess one of the questions should be almost sort of philosophical, which is, what do you believe the role of amyloid is in Alzheimer's disease? So one can postulate maybe three simple models. One is that it triggers the disease process, and after that trigger, it doesn't matter what you do to amyloid because the disease is going. Another idea is that there is a threshold amount that's required in the brain to stimulate, as you suggested, tauopathy. And maybe a third avenue is that it's driving the disease always at all times. So what's your view? Because it's very important in terms of when you administer an anti-A beta therapy as to what effect you think it will have.
Yeah, it's quite a difficult question, I may add here, because I think there are many things we do not really know today. However, what we do know, basically from genetics, that number one, A beta is involved, and in fact, recent clinical data finally show that this is the case. The question is it first? What is actually first? What's coming first? And I can assure you we have many debates in our own company, what is in fact coming first. I think the data, the recent data, in particular with the tau imaging actually, and the data by Keith Johnson at Harvard Medical School really show that there is a very tight interaction between A beta and tau, and that really A beta is driving the tau pathology. That A beta is there and reaches very, very quickly a plateau, and then the spreading of the tau across the brain is actually happening. So you can really consider A beta as a disease which is mediated by or which is mediating with the pathology, meaning the tau pathology in Alzheimer. So taking away this continuous trigger, with continuous stimulus, might actually help to interrupt the tau pathology. And of course, and what we all see, and in fact this is the best explanation, you have to go early, because if you have achieved this plateau, you might not be as efficient anymore in stopping this pathological process. So all points towards, yes, A beta plays a role. It's probably working via tau, and the earlier you take it away from the process, the more chance you have to really have an impact.
Okay, and I'm going to ask you another difficult question, but and keep the response as brief as you can. As you go earlier in the disease, clearly the amount of cognitive benefit that you'll be able to give to patients reduces because they will have mild disease. So the window for efficacy becomes constrained, and that's just the nature of going early. You mentioned the role of the regulators in drug development. Do you think that going further down the line, reimbursement for compounds that will only and can only show a modest effect early on is going to be an issue for the field?
I think if you can interrupt really the disease process before the patients actually need all these expensive drugs, which would be probably the best way of using the money of society. And what again comes out now very heavily is that you even can detect certain symptomatic changes, molecular changes, even before you have a disease, meaning a pre-disease, pre-symptomatic level. For example, tau, and I was again very impressed by the work at Harvard. They actually show in healthy people, healthy volunteer, that if you have tau early on in life, you have no Alzheimer's disease symptoms. You still have tau, and this presence of tau actually makes you much more predisposed to getting the disease. So actually going very early, even pre-disease, and identifying if you have a tau or not, might actually help to identify the patients and potentially initiate preventive measures.
Thanks. Tony, coming onto you now. I think Franz Kafka wrote a book where someone woke up and found they'd been turned into an insect. And I'm just putting myself as a mouse waking up to find I've been joined to another mouse. I just wondered, did you joking apart, did you look for the effects on stress on those animals as a consequence of waking up and finding you have a partner that you didn't really know about?
Yeah, Gregor Samsa. I remember we had to read this book in school. Yeah, I'm not sure if they feel like Gregor when they wake up, but we actually are doing studies to look at stress in the pairs, and we find the level of stress is not different from, let's say, a surgery where you implant, an optic device or anything else, or you inject a virus. The stress level is initially high and then it goes down. We have measures to look at behaviors such as building a nest, and after a couple of weeks they start building nests again, and they just walk around together, obviously. We can even train them to run on a rotor rod, and they get pretty good at it. So it's quite remarkable.
I mean, the potential of your work is just tremendous. I think we all accept that. One of the things that occurred to me is that if you look, really it's the role of epigenetics in this. So if you look at people who are the same chronological age, when you look at their biological age, and sometimes this is very obvious, they seem to be very different. Are you planning to do any investigations into that?
Yeah, that's a really fascinating question. And this is really actually how we got into taking this approach that I showed you. When we look at these hundreds of proteins and take those factors that are most strongly associated with aging, we can predict the relative age of a person. We can call this the biological age, although we don't really have proof that it is that. But then you see these individuals who have they look biologically younger than their time they lived. And I showed you these two individuals that have these high levels of CSF2, for example. Are these individuals, do they have more of these beneficial factors than their peers? And does that maybe make them live longer? And vice versa, a person who has less, are they predisposed to a disease such as Alzheimer's or other age-related diseases?
Okay, thanks. Nancy, just coming on to you finally. It was great to see another target being worked on, I think, for Alzheimer's disease and for dementia, because we have a paucity of targets. I just wondered, is there an A4 knockout, and what sort of phenotype does that have? Because you might anticipate that would have beneficial cognitive effects.
Yeah. So FA4 knockout indeed has been generated, but FA4 itself plays a very important role during development. It is exon guiding factor, and also it regulates synapse formation and maintenance. So it's very difficult to analyze the phenotype using the general knockout. So we know from our studies that if we specifically knock down FA4 in the hippocampus region, it can regulate synaptic plasticity the way that we presented. So it is a negative regulator. In the adult, it certainly has no other effects, unlike in development.
Okay.
Yeah.
And another, I guess, technical question. When one looks at the human disease of Alzheimer's disease, often it requires a lot of neuronal loss actually before, and tau pathology and A-beta pathology, before people actually show any symptomatology, so the brain seems to be very plastic. In terms of interpreting the mouse data where you have A-beta deposition, but often without neuronal loss, how do you translate the findings that you get with your compounds into the disease from that mouse model that actually normally does not have neuronal loss? Is that an issue for you?
So what we're looking at is the early stage, so it's at the synaptic dysfunction stage prior to neuronal loss. So we are manipulating the events at that stage and see how it can impact on the cognitive performance. So I think our data is consistent both at the mechanistic level and also at the behavioral level, that if you can somehow overcome the synaptic impairment, then you can have a beneficial effect on the behavioral outcome.
Yeah, let's see if there are some questions from the audience.
What is the role of glutathione and precursor of glutathione in protection of the oxidation and the aging mechanism of cells?
Well, thank you for your question. I mean, I think glutathione is a recognized antioxidant, but the problem is that if you really want to use it, for example, for disease treatment, then it might not get into enough in the brain. In fact, it doesn't get in the brain at all. So when you think about glutathione and neurodegenerative diseases, then it's not a target which you can use.
Yes, sir.
Thank you, guys. Thank you. It was very good talks. A simple, maybe a comment or solution in regards to slowing down cognitive decline. There was a study in China that was done in looking at bilingual people who spoke more than one language, and they showed that the more languages you know, the slower the cognitive decline. So maybe as a early prevention technique, then learning more than one language is actually a good treatment methodology. Now I'm curious because you show in the U.S. there's a lot potentially more cognitive decline, and actually the average, being very serious about it, I mean, yeah, it's funny, but the average American only speaks English, and the average American probably barely knows proper English, right? But as opposed to, and no offense to my American colleagues, and I'm American, so, but the average European knows more than one language. So this is just maybe something to think about, and maybe you guys can comment on that.
I can comment a little bit on that if the panel don't mind. I think what we're looking at here is cognitive reserve rather than protection against pathology. So when you look at risk factors for dementia and preventative behaviors, the thing that comes out, although this is epidemiology and so it must be treated with caution, the thing that always comes out as being significant is education. So that is a protective factor against dementia. But it's really, I think, that people increase synapse density and they increase their plasticity so that they can withstand the pathology as such, rather than it preventing the pathology. But clearly, and you know, so remaining socially engaged, speaking more than one language, actual professions that have a high degree of analysis and technical elements, these are all protective factors as well. And indeed, if you look at some populations, and one in the UK which has been studied over a generation, it looks like the incidence of dementia or the prevalence has actually decreased somewhat. And that is probably due to a couple of factors. One is that the educational status has increased in that time, but also the advent of better cardiovascular control so that there is fewer vascular episodes and vascular dementia.
The idea of a foreign language as a drug is actually kind of seductive anyway. So. Last question, sir.
In physics, we use infrared spectroscopy to address some of these issues. And in the case of Alzheimer's disease, we see that there's a conformational change of the protein. So the alpha helix opens up into beta sheet, and the Van-der-Waals interaction is much stronger in beta sheet, and that's why plaques are formed. Is it known what triggers this conformational change, and can we block it? Because that could be one type of medication.
Yeah.
It's a great question, and I don't think it is known what triggers it. And if you look at the concentration of A-beta in the brain, in the parenchyma and CSF, it's actually never there in sufficient concentration to spontaneously aggregate, which it will do in the test tube. And so that is a mystery. And if we understood that, there may be a drug target in it. There have been a number of attempts, and I tried this for a long time in my career, to find molecules that prevent aggregation. And in fact, there is a compound in phase 3 trials that is proposed to be an anti-aggregant for tau. Personally, I think that that's a very challenging approach to take for thermodynamic reasons, in fact. Getting enough compound at the site to prevent that process, which is highly entropic in nature, is challenging for drug discovery. Tony.
Yeah, maybe just, it's well known that inflammation can trigger aggregation of proteins, amyloidosis. In fact, before we had mouse models that overexpress APP and other genes, we would induce inflammation to produce amyloid deposits. They were not consisting of A beta, but other proteins. So there is something about the immune response, inflammatory response, that seems to stimulate protein aggregation.
Let's wrap it up here. Thank you. Thank you all.
Thank you very much.
Thank you.